So once air passes through the trachea, it reaches the bronchi. The bronchi has several layers. It's similar but not exactly the same as the trachea. Starting with closest to the lumen is the mucosa, comprised of the epithelia, same subtype, plus the lamina propria. Underneath that, there's a smooth muscle layer called the muscularis. Underneath that, there's the submucosa, similar to what we saw in the trachea, and a cartilage layer as well. However, the cartilage here in the bronchi is comprised of discontinuous plates. So you might see a few, 2 to 3, or sometimes just 1 in the smaller bronchi in this cartilage layer. And outside of the cartilage layer is the adventitial layer, the same as what we saw in the trachea. So what epithelial types are present? Essentially the exact same ones that we saw in the trachea. Once you leave the bronchi, you lose all the cartilage. That actually defines what a bronchiole is. So you leave the bronchi and you enter the bronchioles. These are airways that are about 1 millimeter or less in diameter. No cartilage is present. However, they have a good layer of smooth muscle around them. So what are all layers here? Mucosa, pseudostratified ciliated, and in the largest bronchioles it will be columnar, but as the bronchioles get smaller and smaller, it might be low columnar to even cuboidal in the very smallest bronchioles. The lamina propria here is quite scant. There's not much of it. It's a thin layer. And there's that layer of smooth muscle pretty much right underneath the mucosal layer. And it's this smooth muscle that contracts and can regulate the airway diameter. The smooth muscle can contract and stay contracted, and in that case, that's what happens in asthmatic attacks. So outside the smooth muscle layer, you have adventitia, which is just the connective tissue layer. So what epithelial cell types are present here? Ciliated cells, and as you move down the tree, the respiratory tree here, they become fewer and fewer, as well as their height becoming shorter and shorter. You have goblet cells present, and you have a cell population coming in that we haven't seen before, called the Clara cell. And then you have those granule cells that we mentioned, which are the enteroendocrine cells, and the stem cell population. So what are Clara cells? Clara cells are found usually in the smaller bronchioles, and these are non-ciliated cells that have a dome-shaped apical surface. Their function is to produce and secrete a lipoprotein that acts as a surfactant. This surfactant essentially keeps bronchioles open so that they don't collapse, because they don't have cartilage to keep them open. Clara cells also secrete something called CC16, or Clara cell protein, which is a diagnostic marker in lung injury. So let's take a quick look at COPD, or chronic obstructive pulmonary disease. COPD and asthma are a combination of conditions that result in a decreased gas exchange between blood and air. So it's extremely prevalent in our population, more than 12 million people in the US, and the fourth leading cause of death. It's characterized by obstruction of the airflow. So if the air can't get through the airways down to the place where gas exchange takes place, you have a big problem. You're going to have decreased gas exchange. A person just cannot get enough oxygen in. So that's the good reason for looking at what are the structures where the air is flowing, what are the airways and how do they function, and what happens in dysfunction. So here's a normal lung CT scan. What about this one? Well, in this CT scan, you can see some enlarged airway structures. several of them actually. Why are they expanded? That's called bronchiectasis, by the way. Well, there could be something in the airway, such as foreign objects or mucus. There could be fibrotic buildup from the walls of those airways. You could have a tumor growing there. So all of those things would end up expanding the airways. But the main culprit is mucus. This is a big problem in COPD. So what is the function of mucus? How do we usually make it, and how do we clear it? Well, it acts as a protection in the airway. It moistens the airway. As I said previously, it traps particles and pathogens because it's sticky stuff. And it also contains anti-inflammatory and antibacterial compounds, as well as the mucin proteins, which lend viscosity. Where does it come from? Well, epithelial cells and subepithelial glands all make mucus. So remember the goblet cells that live in that epithelium, they're making lots of mucus, but there's also glands in the lamina propria that have ducts that lead out and also produce more mucus that keeps this whole system lubricated and working. So here's an image of a normal bronchiole on the left and a not so normal bronchiole on the right. What's wrong here? Well, the airway is not patent. It's full of something, and in fact, it's full of mucus. And you can also see some cells that have joined into that exudate, probably from desquamation across the epithelial surface. So let's take a quick look at how normal mucus secretion occurs, and then we can look at a couple of examples of what might go wrong. So normally, goblet cells make mucus constitutively. That means they're always making some and secreting it as it goes. All right. They also can secrete upon stimulation. So imagine that you ate something really spicy. All right. The sensory receptors in that mucosa would get set off, a signal would be sent in, and it would send back saying, secrete some, we have to clear this stuff out. All right, so then, you know, your nose begins to run because you ate like a really spicy taco or something, right? So this diagram is upside down. The airway is at the bottom here, right? This would be the layer of mucus here, and these are the cilia for the ciliated cells, and then this is the mucus. So secretion is going in this direction. So what goes wrong? Well, inflammation can get started from a variety of sources. You can have infection, you can have allergy, you can have environmental irritants like cigarette smoke or dust or something like that. And all of those can alter mucus production. Essentially, interleukin comes into play here, and it stimulates either hyperplasia of goblet cells, which means you get too many goblet cells, hyperproduction of mucus in the existing goblet cells, or outright loss of function of the cilia. There's also genetic causes that lead to mucus blockage of the airways, called primary ciliary dyskinesia and cystic fibrosis. We'll take a quick look at all of these. So in interleukin-stimulated hyperplasia, the inflammatory source actually affects the T lymphocytes, which put out some interleukins, and those stimulate the goblet cells to undergo mitosis. So you get an increase of the number of goblet cells there. Then you have too much mucus being secreted into the airway, and you can get blockage. Interleukin-stimulated hyperproduction works in a similar way. So whatever the inflammatory source is, it stimulates those T lymphocytes, and a different interleukin stimulation mechanism is engaged, and the existing goblet cells start producing kind of in overdrive. So you don't get an increase in the number of goblet cells, they just start really overproducing. And again, all of that mucus that they're making gets sent out into the airway and can block the airways with mucus. In a similar fashion, an inflammatory source can stimulate the production of interleukins that actually interfere with the cilia themselves. So certain interleukins can stop the cilia from working properly. And if the cilia don't beat, it doesn't matter whether the mucus is the right amount or not, it's not going anywhere. So that mucus then does not get moved up the airways and out as it should normally. So not only do you get mucus blockage possibilities here, you can also have a lot of infection, because if there were trapped pathogens, they could then replicate and multiply in that nice cozy layer of mucus, and you could get bad things happening from that. So here's a specimen from an obstructive airway patient. You can't really tell much there. You're really just looking at the cilia in the ciliated cells. All right, so you're looking at the actual cilia profiles. When you look at higher mag, though, you see there's something wrong here. This doesn't look like the normal 9+2 cilia arrangement. And there's another problem with it as well. It doesn't have the right number of dynein arms on each microtubule doublet. You're supposed to have 2 dynein arms, and in this particular patient there's only 1 dynein arm. The inner dynein arms are actually missing, as well as the doublets being disarranged. This is what it should look like, and you can compare it to the other picture there, and it just is not organized properly, and you can imagine that it wouldn't function properly. So this is called primary ciliary dyskinesia. This is when the problem with the cilia is genetic. So you can have an acquired version of it where the interleukin causes problems with the cilia function, or you can have a congenital defect where the dynein arms are completely absent or partially absent, and again, the cilia don't beat, and if the cilia don't beat, the mucus doesn't move. Another problem here is that if cilia don't beat because there's no dynein arms, then the person, if they're male, is probably also going to be sterile, because the flagella of the spermatozoa don't beat either, because they're essentially just long cilia. So those sperm aren't going anywhere, and so somebody who has primary ciliary dyskinesia will have numerous respiratory infections, as well as, if they're male, being sterile. So cystic fibrosis is another condition in which the mucus can block the airways. In this case, it doesn't have to do with the cilia or overproduction. It has to do with the viscosity of the mucus. So the altered chloride channel, all right, leads to increased mucus viscosity. In other words, it's too thick. So no matter how hard those cilia work, they can't move that thick layer along and up out of the airways, and you get mucus stasis and blockage.